Longevity medicine aims to slow aging and reduce the risk of age-related diseases through therapeutic and lifestyle interventions. While still an emerging field, there is growing evidence that certain strategies can positively influence longevity. Case Study 1. Calorie restriction in rhesus monkeys. One of the most well-established methods for improving health and lifespan in various species is calorie restriction (CR), whereby daily calorie intake is mildly but sustainably reduced. A landmark primate study provided some of the strongest evidence yet that CR may work in humans as well. Beginning in 1987, the National Institute on Aging initiated a long-term project in collaboration with University of Wisconsin studying the effects of CR in rhesus monkeys, our closest animal genetic cousins. One group received normal diet while the other had calorie intake reduced by 30% from youth onwards. After over 30 years, the CR monkeys showed notable benefits. Their risk of cancer, cardiovascular disease, brain atrophy and more was dramatically reduced compared to controls. Furthermore, CR monkeys lived 10% longer on average and showed fewer signs of frailty at equivalent ages. This seminal research proved that longevity interventions translatable from model organisms could work for other primates as well, and opened doors for further CR studies and mimicking strategies in clinical trials. It demonstrated aging's flexibility despite evolutionary conservation across species. Case Study Number 2. Anti-diabetic drug metformin. Metformin is a commonly prescribed oral anti-diabetic that has generated much interest as a potential anti-aging medicine due to interesting patterns observed from its widespread use over decades. There is compelling evidence it may help prevent and treat age-related diseases beyond just diabetes. Large population studies have correlated metformin usage with reduced cancer incidences, lower cardiovascular mortality, less frailty, lesser cognitive decline, and longer lifespans in diabetic patients versus those not taking it long-term. There is growing consensus it activates similar pathways in the body as calorie restriction for its pleiotropic benefits. Several ongoing clinical trials are now systematically exploring metformin's dose-response effects on aging parameters, metabolic health and aging-related disease biomarkers in healthy elderly populations versus placebos. If proving successful long-term, metformin may offer a safe, inexpensive approach to mimic benefits of calorie restriction without needing dietary changes difficult for many to sustain. It represents a promising strategy translatable to wider usage based on existing safety data. Case Study Number 3. Naked mole rat. Lessons with lifespans over 30 years. Naked mole rats are the longest living rodents and exhibit almost no signs of aging till late in life. This phenotype has profoundly advanced the science of longevity by offering novel insights into anti-aging mechanisms from study of their unique biology. Key lessons learned helpful for human therapies include hyperactive DNA damage response, proteostasis maintenance through mole rat-specific chaperone proteins, suppressed reactive oxygen species accumulation, and extreme resistance to tumor development despite multiple insults causing cancer in other rodents. Researchers are now exploring how to recapitulate mole rat traits beneficially in humans by activating similar pathways through drugs without adverse effects. Several small molecule or peptide therapies entering clinical trials aim to improve DNA repair, enhance protein homeostasis like proteasome peptides, scavenge toxic waste, or directly stop tumor formation, inspired by naked mole rats. Understanding this remarkably long-lived species has opened novel strategies targeting hallmarks of mammalian aging not apparent from model organisms before. It expands the scientific toolkit against disease and disability with age. Case Study Number 4. Senolytics to clear senescent cell. Cellular senescence refers to a permanent growth arrest state that limits replication of damaged cells but also has negative consequences when they accumulate with aging. This realization drove development of senolytic drugs intended to selectively clear senescent cells from tissues as a novel anti-aging approach. Initial success was demonstrated in mouse models of atherosclerosis, pulmonary fibrosis, frailty, and more, where senolytics proved capable of reversing age-related diseases or dysfunction by simply targeting these pro-inflammatory cells. This inspired first-in-human clinical trials using two types of senolytic compounds. A pilot study clearing senescent cells from an idiopathic pulmonary fibrosis patient showed safety and helped halt disease progression. Larger trials are now ongoing evaluating effects on cardiovascular aging. biomarkers, metabolic health, and pulmonary fibrosis when dosed to older adults. This work confirms senescent cell accumulation as a druggable target of aging. If human results parallel success in rodent models, senolytics could transform treatment of common age-associated inflammation-linked conditions resistant to existing therapies through a relatively straightforward approach, proving highly impactful. It exemplifies how longevity research is yielding novel pharmaceutical strategies. Case study number 5. World's oldest man. Among the strongest real-world proofs longevity interventions can work are exceptional longevity outliers living well into their 110s while remaining active with sharp cognition. One symbolic individual who captured global attention was Shigechiyo Izumi, certified by Guinness World Records as the world's oldest man in 1986 at the age of 120 years, 259 days before passing away in 1986. Izumi attributed his remarkable health span to an active lifestyle spent farming rice well into his 100s, plus a plant-rich Japanese diet heavy in isoflavone-rich soy, fish, and vegetables, but limited meat. He engaged socially, exercised regularly, and never smoked or drank, habits promoting maintenance of metabolism, immunity, muscle mass, and mental sharpness, even at the oldest ages, unlike many peers. His case showed that optimal environment and behaviors can help push the boundaries of human aging. Potentially heritable traits may have contributed as well, given 3 siblings also lived well into their 100s. Izumi lived independently, with good appetite till the end without major diseases despite extreme age. He inspired hopes that optimized wellness habits combining elements off blue zone regions worldwide could enable more people to stay autonomous into their 110s as lifespans continue lengthening through medical and public health gains. His achievement strengthened belief aging's flexibility is underappreciated given the right environment. Case study number 6. Longevity athletes breaking records. Contrary to common perceptions, advancing age does not preclude athletic achievement or physical fitness, as exemplified by several outstanding senior athletes setting new age group records. Their sustained health, stamina, and competitive spirit demonstrate practical benefits of adopting a lifelong, endurance-oriented lifestyle aligned with longevity medicine principles. For example, Fauja Singh ran his first marathon at age 89, then set world records in the 1:00:10 for age bracket from 2011 to 2013 as the oldest person ever to compete in the event. At 95, he still ran 5Ks. The late Melanie Blattel also set records racing triathlons into her 70s, while Helen Klein ran marathons into her late 90s, winning world titles above 90. On the extreme end, 95-year-old Eddie Pinder lifted weights competitively, holding world bench press records. Their longevity results from diet, lifelong aerobic activity, keeping metabolic and cardiovascular parameters youthful as supported by body composition, blood tests. While their exceptional genetics can't be underestimated, chronic exercise truly reduces biological age. This inspires others that age guidelines restricting seniors are too rigid if health allows, and moving our bodies has direct benefits on the rate of aging, not solely youth-focused fitness goals. It highlights exercise as a key modifiable factor for maximizing disease-free life years. Case study number 7. Lifestyle optimization effects. Some remarkable individuals demonstrate longevity medicine's potential through sustained multi-pronged lifestyle customization alone, achieving extreme health spans without drugs or biotechnologies still in development. Their longevity owes to taking ownership through rigorous self-experimentation. Perhaps the ideal embodiment is William Longo, professor who just turned 104, but maintains a body composition, activity levels, and cognition better than most 70-year-olds through precise optimization. He strictly follows a low-sugar, plant-based whole food diet, fasts regularly to recycle cells, gets adequate micronutrients based on blood tests, practices yoga daily, maintains social engagement, and adheres to extended water fasting 1-X yearly to trigger cellular stress resistance. Proven anti-aging interventions synergize for maximal effect. While far harder for many to sustain over 100+ years than simple pharmacological approaches, persons like Longo exemplify that an extreme health span exceeding average human lifespan is achievable currently based on lifestyle personalization alone and desire to continually improve well-being without relying on unproven treatments. They motivate rigor in applying multi-dimensional strategies, balancing bodily systems for success against Father Time himself. In summary, these diverse real-world examples, from human to animal studies, illuminate the breadth of approaches investigated within the promising field of longevity medicine and proof it is transforming aging from an inevitable process into one we can proactively shape. From dietary changes to drug repurposing, stem cell regeneration to simply clearing toxic cells, the Longevity Toolkit continues expanding through dedicated research aiming to keep minds and bodies vigorous at unprecedented ages. With time, many of these strategies are poised to become widespread, having proved safety, and some are already aiding lives today. Continued progress holds tremendous potential to revolutionize what's possible for the future of human health.